So let's talk about the first um, group of hormones, that is auxin. So it was first not discovered actually, but proposed by Charles Darwin. You know Charles Darwin? The monkey guy. What? Yeah, yeah. The fathers of modern evolution. Yeah, right. Um, I during my intern, you got to do intern. The intern. I did like 2 weeks my intern work in Charles Darwin home in what's the name of that county? Kent. His home has got a home. Anak dia ramai, anak dia 10, so the home is so big. So he did a simple experiment. I'm going to show you, and he noticed that if you allow the seedling to germinate, to grow, when there is a light coming from one side, like in here, the coleoptile means the protective sheath of the plumule, will start to grow towards the light. When the tip is removed, it stays erect. When the tip is covered with something opaque, it's going to stay erect as well. But when the tip is covered with something transparent, it follows the control. When the only the base is covered, it will follow the control. So he started to hypothesize, postulated that there must be something, some kind of chemical or substance that get produced within the coleoptile region, the tip region. Okay, and then of course he died. I think when he died, he got buried, and he's now neighbor with Newton, the same place. You know Newton? Did you learn calculus? That's from Isaac Newton. They are neighbor now. Betul, kau tak percaya? Kau tak percaya? Kau cari benda ni. I think this is where they are buried. Bukan Abbey. Westminster Abbey. You know London got the clock, the Big Ben? Okay, next to it. Westminster Abbey, next to it. Belah aja. Very close. It's like here, and then you go to Dean's office. Right. Okay. So, and then the other guy continued the experiment, and then between the coleoptile and the main body of the seedling, he separated with two kind of blocks, the gelatin block and the mica block. The gelatin is porous. It allows whatever compounds, if ever present in the tip, to go down. And when he used the gelatin block, the plant, the coleoptile, is following the control. But when he used the mica block, mica is pretty much is a mineral, like a stone. No response whatsoever. So now they are confirmed there must be something in the coleoptile. It's not in any other regions that control this phototropism for the seedling to follow where the light is. Okay, and then when the time progresses, the scientists, so this is where you can appreciate the work of scientists. It's not the work of one guy, one scientist. It's a continuous work. This is why I keep stressing on a good scientific conduct. Because whatever your discovery, whatever your observation, it's going to be useful somehow down the line by other scientists. Okay, so the other scientists isolated, meaning extracted the compound from the coleoptile tip, and it's named Auxin. From the Greek, Auxin means to grow. And the form of the Auxin, remember there are many of Auxin that got extracted in the form of IAA, indoacetic acid, generally produced by the growing apices of the stems and roots. See, stems and roots. These are two completely opposing organs of the plant, end to end. One at the end of the shoot system, and then one at the end of the root system. You don't get this in humans. Okay, you don't get your pituitary is produced in your pituitary gland. Your pituitary got produced in your finger, in your hair. No, no. Your hormones pretty much produced by a specialized gland. Your pituitary gland is not going to produce estrogen. What happens if your pituitary gland producing estrogen? What potentially will happen? Meaning that you get estrogen activated all the time, right? If estrogen get activated all the time, you are not able to grow properly. Okay, you will start to look funny. You'll feel funny, and more or less, you're not going to be human. Yeah, yeah, yeah. So is estrogen is a bad thing? No, estrogen is not a bad thing. It has to be around in the right time, in the right amount. Okay, that's why in human it's specialized. Right. So what? And then scientists wonder again, what's with the bending? I mean, like, if in the whole cylinder of the coleoptile, is there any specific part of the cylinder is actually producing the auxin? Is it here? Is it here? Or is it here? So the scientists started to think about this because they hypothesized that in the cylinder region of a coleoptile, there must be one region that is producing auxin because they believe that localized knowledge from the human. Because a human, you got the gland, right? So since coleoptile do not have gland, there should be some regionalized production region for the auxin. So what this scientist did was, he cut the auxin and he put it in the agar to transfer whatever auxin that is present in the coleoptile into an agar block. So this agar block now contains auxin, and then this auxin is placed on the decapitated coleoptile, coleoptile A and coleoptile B, in a rather asymmetric placement. It's not completely on top of the cut head, but just on the edge, in this manner. And they see that when it's placed this way, the bending is occurring on the shorter side where auxin is not placed. So now the theory is confirmed. Auxin is actually present when the plant is bending in certain way. The longest curvature is where auxin has to be the most. Right. So of course, the experiment got further proven, and they cut it, they put it on either side now. No matter which side it's putting, whether it's this side or this side, the curvature will be on the opposite direction. If the auxin is placed on this side, on this edge, the curvature will go the opposite way. If it's placed on this side, the curvature will go on the opposite way as well. So what's with that? Why won't it form the curving according where the auxin is placed? We'll see in a bit. So it turns out that later it was found that the auxin is actually shying away from the light. Okay, so when a coleoptile like this is facing the light, auxin actually shying away or moving away from the light. When this happens, the darker side of the coleoptile will be concentrated with auxin, and this is why the longer curvature happens on the darker side, while the side that is facing the light will curvature or curving the leaves. And that's why you got the bending towards the light source. And this is the explanation for why phototropism is happening. Okay. Why auxin is shying away from the light? And why is that? This is the nature of auxin. Okay. There are many compounds that are actually sensitive to light and temperature. For example, vitamin C. You see, if you cook your vegetable and fruits for too long, above 70 degrees, that's going to destroy vitamin C. That's why it's not healthy to eat fried fruit all the time, baked, roasted, satay, that kind of stuff, because definitely no vitamin C in there. Why do you think after you have eaten satay, people give you all the onions, the cucumber and so on? The wisdom is there because there's no vitamin C left in your satay. So you got replenished for your antioxidant by eating all these salad cubes. Burning your meat is not a healthy way to eat it, but people eat it because it tastes so damn good, right? So to compensate, to neutralize the free radicals that's happening in your body, eat more salad. All right. Auxin, there are two types of auxin. There are the native auxin, meaning that you can find this auxin naturally in plants. You can have the synthetic form produced in the factory, but in plants we got this. We got the IAA, indole acetic acid. I do expect you to know this, okay? So at your level, when people want to ask you which auxin are you using, you need to know what kind. Is it IA? Is it IBA? Is it NAA? Okay, right. And then you got the IBA, butyric acid, and then phenylacetic acid, PA. And then you got the synthetic auxin. Okay, these synthetic auxin, they have other functions than stimulate growth in plants. They can be used as herbicides. Have you taken your wheat science?
No.
In wheat science, later you will learn there are many types of herbicides. And some of these herbicides, actually, they are auxin, the synthetic auxin. Okay, you got 2,4-D, alpha NAA, dicamba, and prodon, picloram. There are many types.